Welcome to the video for chapter 10.2. We are now going to start talking in more detail about our different superphyla. For 10.2, we're going to focus on our gram-positive phylas. So this is going to be our Firmicutes and our Actinobacteria. So our section objectives for 10.2, you're going to be able to describe the traits that enable Firmicutes to survive dryness and persist for long periods, aka they make endospores. And then finally, you're going to be able to describe how diverse actinobacteria have both positive and negative influences on human health. So this is where we're going. Let's talk about some specifics. Okay, so let's talk in more detail about our Firmicutes and our actinobacteria. For the most part, these are bacteria that stain gram-positive, meaning they're going to be purple after the gram stain. Firmicute literally stands for tough skin, which is very appropriate because there's a large amount of bacteria underneath this phyla that can create endospores. And we know that endospores are both heat resistant, chemical resistant, antibiotic resistant. They pretty much can survive about anything. So tough skin indeed. Now, they are characterized by having a really thick cell wall. Okay, this is going to help them resist drying out. They are going to have several layers of peptidoglycans supported with teichoic acids like we talked about when we talked about cell envelopes. Now, one of the things that's really important to understand is actinobacteria also stain gram-positive, so they also have a thick peptidoglycan layer. Now, a lot of actinobacteria are going to have an additional waxy coating on the outside of their cells, which is going to help them avoid things like our immune system. Now, one of the ways that we can tell the difference between Firmicutes and Actinobacteria is by looking at their DNA sequencing. So in Firmicutes, they have low GC content, which means that compared to adenine and thymine, the amount of guanine and cytosine in the DNA is considerably lower than what it should be. And Actinobacteria are the opposite. They have extremely high GC content, meaning they have very little A's and T's in their DNA. Now, this is a table from section 10. These are going to be a few examples of Firmicutes and Actinobacteria that we might be familiar with. So let's talk about our Firmicutes. We have talked about several of these so far. We have Bacillus anthracis. This is that soil bacterium that's going to cause anthrax. We have Bacillus subtilis, which is also a soil bacterium. We use this a lot in research, just like we do E. coli. We have C. diff. We've talked about this. Like if you have really strong antibiotics and you don't eat your yogurt or probiotics, you can kill all the good bacteria in your gut, leaving C. diff, which is the bad bacteria. Okay, this is going to cause a lot of GI issues. It's really actually really terrible, and it's really hard to get rid of once you have it. We have Clostridium botulinum. We talked about this. This causes botulism. It's used to paralyze people's faces to get rid of the wrinkles in Botox. We have Clostridium tetani. This is going to be the bacterium that causes tetanus. So all of these have the same characteristic. They're all endospore formers, so that they can become an endospore and survive bad environments. Now we have the non-spore formers for the Firmicutes. This is going to be things like Lactobacillus and Lactococcus. Now, Lactobacillus and Lactococcus are used for dairy culturing. They're used to make yogurt and cheeses. In fact, Lactobacillus and Lactococcus are one of the main ones that are listed on your yogurt container. Okay, so they're good bacteria. We have Enterococcus. This is going to be an intestinal bacteria. We have Listeria. This is a pathogen. It's an intercellular pathogen, so it tricks its way into your cell via endocytosis, and then it hijacks the endosome instead of having it merge with your lysosome and kind of degrade everything that was brought in the endosome. It prevents that from happening and makes the endosome its new home. And then it can use the endosome to travel from one cell to another and spread the infection. Listeria, we're going to talk about this one because it's kind of scary, actually. We've talked about Staphylococcus aureus. This is a skin infection. It can cause toxic shock. But we talk about more as, because it's one of our escape pathogens, we're more familiar with MRSA, the methicillin-resistant variety. We have Staphylococcus epidermidis. This is the same as the one that we use in the lab all the time. So this is a common skin microbe. Now, one thing that we haven't talked about is a lot of the firmicutes are gram-positive, but there are some members of this firmicute phyla that have done something weird. They have lost their cell walls, so they don't stain gram-positive. So we're going to have to do things like acid-fast stain in order to visualize them underneath the microscope. Okay, we call these the molecutes. These are our mycoplasmas. This is Mycoplasma genitalium. Okay, so this is going to be found in a lot of genital regions. And then we have Mycoplasma pneumoniae that causes pneumonia. Now, because these lack cell walls, they don't gram stain purple. They don't gram stain at all, actually. So we're going to use that acid-fast stain to view them. So let's talk about Actinobacteria. We divide this up into two categories. We have the Actinomycetes. These are going to be, we're going to be our antibiotic producers. And then we have other Actinobacteria, the ones that don't produce antibiotics. We've talked about a few of these. Cutibacterium acnes. This is the bacteria that causes acne. We have Micrococcus luteus. When we did the isolation streaking, that yellow bacteria that you separated out, that was actually Micrococcus luteus. We have our mycobacterium, so Mycobacterium leprae and Mycobacterium tuberculosis. These ones are going to not gram stain as well because they have a thick waxy capsule on the outside of them. They're going to actually be viewed with the acid-fast stain, like the molecutes. Okay, and then we have this last guy in the Actinobacteria phylum that we're going to talk about. This is Propionibacterium Freudenreiche. My German's terrible, so I probably just mispronounced that. But this is the bacteria that makes Swiss cheese Swiss cheese. This is what's going to create the holes in the cheese and that really tangy flavor that it has. Okay, so let's talk a little bit more in detail about some of these bacteria. Okay, so now that we've talked about some of these bacteria that we have talked about in the past and you might be familiar with, let's get into some more details, specifically things that you need to know for the next exam. Now, one of the things that make firmicutes famous is the fact that there is a large contingent of them that form endospores. Okay, remember endospores are heat, freezing, chemical-resistant spores that can live in the environment and remain inert for thousands of years until conditions get better and they can become viable bacteria again. Endospores are very concerning both medically and in the environment. We just can't get rid of them. In fact, the only way that we can ensure that we don't have endospores in things like our food or on medical instruments is to use an autoclave in order to use pressure to drive the temperature up enough that we can actually get rid of the endospores or the bacteria that forms the endospores. Now, when we're talking about endospore forming species, the one that we're going to talk about the most is the Bacillus species. Okay, Bacillus is found in the environment and it's actually very important economically. So Bacillus anthracis, this is the source of the disease anthrax. It's a very toxic soil microbe. If you remember back in the early lectures, we looked at that picture where that big black mark was on that rancher's neck. That's the source that Bacillus anthracis can cause. Now, we don't have a lot of naturally occurring cases of Bacillus anthracis. It's not very common. As I said earlier, we see it mostly in farmers and ranchers. But there has been incidences in the past where Bacillus anthracis has been bioengineered to be a bioweapon. In the early 2000s, there was a bunch of anthrax attacks that used that bioengineered version of Bacillus anthracis. So we'll talk about that in a few chapters. Now, economically, there is a Bacillus species called BT. Don't ask, I can never remember and can never pronounce the T in BT. But BT is really important is because it's used as a pesticide. Specifically, it's used in worms. So it's detrimental to worms. So it can be used on things like corn, and when it gets into the corn kernels, the worms that are trying to invade that corn actually pick up BT, and BT actually kills the worms off. So it acts as a natural pesticide for us. Okay, so the other endospore forming genus is the Clostridium genus. Clostridium species are obligate anaerobes. They don't like to be in oxygen environments. They also form endospores. So we talked about one of our Clostridium species, Clostridium botulinum, in one of the earlier in one of the earlier case histories. So they are going to have this characteristic club shape. Okay, so they have the area that's going to form the endospore and the vegetative cell portion. Okay, so this is what shows up. Now, we don't really gram stain endospores. We're going to use something like the endospore stain that we've talked about. So you can see here in this micrograph the club shape, and we can see the green and that's going to indicate that the spore is forming. Okay, now we have two really well-known pathogens that come from the Clostridium species. We have Clostridium tetani, which causes tetanus, and we have Clostridium botulinum. We also had one that used to be categorized as Clostridium, but it has a different classification, but it still is a firmicute, and that is C. difficile, or C. diff. This is actually a pretty well-known pathogen. These statistics are a couple of years old. This last year, there's been over 500,000 reported cases in the U.S. hospital system, with about an average of 29,000 deaths a year. So C. diff is becoming more and more prevalent, and C. diff is one of those things that is easily preventable if you follow your doctor's recommendations for the most part. So like if you're on really intense and strong antibiotics, you're usually recommended to eat a bunch of yogurt and to take a probiotic. So what's going to happen is when you're on antibiotics are going to kill off all your good gut bacteria, which is currently keeping C. diff, which is our bad bacteria in our intestines, from running rampant. So when you go on antibiotics, it's going to kill off all your good bacteria, which leaves C. diff with nothing to keep it restrained. So it's going to multiply, and it's going to start to cause a bunch of GI issues. And as I said, C. diff is really hard to get rid of. So you can get rid of it with antibiotics, but a lot of times people never are 100% the same after they get C. diff. One of the best treatments is actually fecal transplants in order to get a good gut microbiome going again. We've talked a lot about botulinum. We talked about it in the case history with Julio and his sister who ate contaminated chili from the can. So Clostridium botulinum makes a protein toxin that is a paralytic, so it shuts down your muscles and being able to use them. We have a lot of people that use it to make themselves look younger, right? This is the whole thing behind Botox. They've taken the botulinum toxin and they have refined it so it doesn't cause disease. So when they inject it into your skin, it's going to paralyze your muscles, which are going to smooth out your wrinkles. Okay. There's forms of infant botulism. They're actually really quite terrible. There's a reason why there's lists of things about when you can feed your child certain things by certain ages and what to avoid to certain ages. There's things like honey that can have Clostridium botulinum in them. If a infant catches botulism from the botulism toxin, it can cause it to grow in their colons, which is going to cause paralysis throughout the infant. And it causes what's called floppy baby syndrome, where their whole bodies are paralyzed and they have no control over the limbs. So they're going to kind of hang there like a rag doll. If you shake them, they have no control and their limbs just flop around. Okay, so botulism's not that great. Okay, so we need to talk about the other side of our gram-positive firmicutes, and that's the non-endospore forming ones. So we have some different rods that don't form endospores. So these are going to be things like lactobacillus. We also have lactococcus, the spherical form of those. Both of these are lactic acid bacteria, meaning they ferment lactose to make lactic acid. So they're going to ferment milk to make milk products, things like yogurt and cheese. In fact, a lot of times yogurt is going to be something that people who are lactose intolerant can eat because lactococcus and lactobacillus and these dairy bacteria have actually converted the lactose into something else. So you're not going to be as allergic or reactive to it. Another one that falls underneath the firmicute phyla is the Listeria species. Listeria is interesting because it's an intercellular pathogen. So it gets into your cells and stays in your cells. It's going to cause disease. It's going to cause GI tract issues. It can also affect your nervous system with the infection. We talked about this a lot. We talked about that case study where the developer for that tech company in Silicon Valley ate a contaminated roast chicken. Okay, one of the things that's really scary about Listeria is a lot of microbes that if you put them in cold temperatures, they stop growing and go dormant. In fact, we store our E. coli and our Staphylococcus epidermidis samples in the fridge and in the classroom in order for them to not grow but be there when we need to use them to make samples for lab. Listeria can actually grow up to temperatures as low as 4 degrees Celsius, which is approximately the temperature of your refrigerator. So they thrive in cold temperatures as well as warm. Okay, one of the things that makes it a terrible foodborne illness is the fact that it does grow at low temps and it does grow really quickly. Okay, but what's really scary is the fact that it will gain entrance into your cells via phagocytosis. Okay, it's going to enter the endosome or the phagosome. Okay, it's going to lyse your phagosome. It's going to start to make more and more listeria. It's going to form these actin tails, which then are going to push onto the walls of the bacteria, the walls of your cell, and this is going to actually encapsulate the bacteria and allow them to penetrate a neighboring cell without ever leaving the cell and being exposed to the immune system. Okay, so Listeria is kind of a scary one. Okay, we also have some gram-positive cocci. These are going to be things like Enterococcus. This is normally in the intestines, but if it gets outside of your intestines, like in your urinary tract, it's going to cause infection. We have Streptococcus. You are probably most familiar with the one that causes pneumonia diseases. So things like strep throat. If you have strep throat, you've been infected with Streptococcus. There's also some form of Streptococcus, like Streptococcus pyogenes, that can actually cause tooth decay as well. Then we have our Staphylococcus genus. Staphylococcus organisms are mostly skin microbes. So we have Staphylococcus epidermidis. Okay, that's a normal part of our skin microbiome. We have Staphylococcus aureus, this can cause staph infections. We're more familiar with the MRSA version of Staphylococcus aureus. This is the methicillin-resistant version. This is one of our escape pathogens. Okay, so we have gram-positive spheres as well as rods. Okay, so scientists are always studying different microbiomes. Like they studied your armpit microbiome, they've studied your intestinal microbiome, your mouth microbiome. One of the really interesting studies that's, really going on right now is there is a group that are studying belly button microbiomes. Okay, so in this study, over 500 individuals have had their belly buttons swabbed and the microbiomes in their belly buttons sequenced. This study has revealed with these 500 individuals over 2,000 different strains of bacteria. And the really interesting thing is that most of the strains that have been identified in this study are most often in the Firmicute and Actinobacteria phylums, which is pretty interesting. Okay, so that's what we should know about our Firmicutes. Let's talk about our Actinobacteria. Now, the Actinobacteria are also sometimes referred to as branching bacteria because some of them look like branches when you look at them underneath the microscope, and we'll see some of that on the next slide. Now, Actinobacteria is a very broad group. We have our antibiotic producers, we have some decomposers that are going to help things break down in the natural environment. We also have some Actinobacteria that are pathogens. Now, actinobacteria are going to have unusual cell wall lipids like mycolic acid. Okay, so they're going to mostly stain with the acid-fast stain and not the gram stain. Now, one thing that's important to know and for your next exam is that the group in the actinobacteria that produce antibiotics are the actinomycetes group. Okay, this is going to be our ones that make our antibiotics. Okay, so this is what I mean by branching bacteria. It really does look like a branch, like not just single bacteria, but a lot. Okay, so even though they're bacteria, sometimes they look like fungi, like different fungi. But these guys are actually our major source of antibiotics. In fact, there's lab groups that go out and literally test soils and take the soils and put them into DNA sequencers, trying to find more actinobacteria that produce antibiotics. Okay, so if we look here, hopefully my pointer's working. If you look here on this left side, you see these really interesting pink and blue and yellow marks in these colonies of bacteria. If you look really closely in these cells, you can see these bluish purple dots. Okay, these are actually liquid antibiotic that the cell is making and pushing out through their cell wall in order to kill off any bacteria of other species around it, and which is going to leave it free to use all the resources in the area. So it's really killing off the competition around it. Okay, we actually isolated a lot of these antibiotics, and we use them in medicine. So what we need to talk about now are the actinobacteria that don't produce antibiotics. So the actinomycetes group, they're the most famous because of the fact that they do produce antibiotics, but we have some equally famous bacteria in the other group that don't. So some common characteristics these bacteria have, they have cell walls that contain mycolic acid. Now, this mycolic acid is going to give this waxy coating, which is really hard for things like our immune cells to hold on to, but it also makes it so that they're not gram stainable. So in order to view them underneath the microscope, we're going to have to use a different differential stain. So we're going to use something like the acid-fast stain that's going to use heat to get the stain into this waxy layer. A lot of these bacteria are going to fall in the pathogen or symbiont categories. So pathogens cause disease. Symbionts are actually bacteria that we have a symbiotic relationship with, which means that we provide a nice, safe, good place for them to live, and they help us out doing other things. So things like some of the bacteria in our guts, they help us digest things like cellulose that we wouldn't normally be able to digest on our own. And they help us get the calories and the energy and the nutrients that we need from those foods that we can't break down. Now, this phyla is going to be composed of both bacilli and cocci, so rod-shaped and spherical-shaped bacteria. Some common ones that are well known are Gardnerella vaginalis. That's a hard one to say, guys. This bacteria is going to cause bacteria vaginosis, which is a vaginal infection. We have things like C. diphtheriae that causes diphtheria. Then we have Propionibacterium. These are the ones that make Swiss cheese. They're going to make propionic acid, which is then going to create Swiss cheese's characteristic holes and tangy flavor. And then we have the Mycobacterium genus. This is going to be the cause of some human diseases like tuberculosis and leprosy. Okay, so let's talk about Mycobacterium a little bit more. These are going to acid-fast stain. They have that waxy outer coating. That waxy outer coating is actually going to do some things that are detrimental for the pathogen themselves. They are very slow growing because of this thick, waxy cell wall. Okay, on our end, it makes them difficult to culture and treat. In fact, both leprosy and tuberculosis, they are both treatable with antibiotics, but there's a lot of cases where they tend to come back because we don't take antibiotics long enough in order to get through that thick cell wall. Okay, so our acid-fast stain is going to stain them purple so we can view them. Okay, so let's talk a case study. This case study is called TB, I'll Be Back. Can't help but hear that in Arnold Schwarzenegger's accent, right? Okay, so our case history is following Michael. Michael is 45 years old, and he's been feeling pretty tired. He hasn't been able to sleep, and he's lost his appetite and has had a very persistent cough for the last several weeks. So he goes and sees his doctor. Okay, so they run a bunch of tests on Michael, but one of the things they do is they take a chest x-ray. Now, this chest x-ray is going to reveal that Michael has a bunch of small granulomas spread throughout the lobes of his lungs. So granulomas are nodes of inflammation or infection. Okay, so if we see here, these wispy parts, so these wispy things are the infection. This is the granulomas. So they're really small. They're about 1 to 5 millimeters. Okay, so when they detected these granulomas, we needed to run a few more tests on Michael. Okay, so they requested a sputum specimen. Now remember, sputum is not just spit, it's both spit and phlegm, so trying to get some of that mucus up. They actually have special booths where you go and give a sputum sample, and we'll talk about that in a few lectures as well. Okay, so his sputum sample had this acid-fast bacilli. So the physician is actually going to diagnose Michael with secondary tuberculosis. Okay, when we have secondary tuberculosis, this means that Michael has had TB before, and it just wasn't all got rid of when he went through antibiotics. So it stuck around being dormant. Okay, we a lot of times also refer to this as latent tuberculosis. Now, one of the reasons why he was diagnosed with secondary tuberculosis is when they were going through his symptoms, you remember having the same symptoms a couple of years earlier, and it wasn't as bad as it is now. So this is why the doctor is going to diagnose him with secondary tuberculosis, because it sounds like he had it before. Okay, so as we know and we talked about a lot, tuberculosis is caused by Mycobacterium tuberculosis, and it's going to infect the cells. Now, tuberculosis is transmitted through respiratory droplets. Because he was diagnosed with TB, Michael is placed in isolation, and in order to get rid of the TB, he has to be on a 4-drug antibiotic treatment for a long time. Okay, it's what we call a long-term treatment. So his 4-drug therapy goes for 2 months, and then so he takes those 4 antibiotics, and then they reduce his therapy to only 2 antibiotics, which are going to persist for the next 4 months. Now, good news for Michael is he responded well to the treatment, which means Michael doesn't have one of the multi-drug resistant strains of TB. Multi-drug resistant TB is actually becoming a lot more prevalent as time goes on. So we're starting to see it more and more often, and there's not much we can do for it. So thankfully, Michael didn't have the drug resistant one, and he was able to make a full recovery. Okay, so this is 10.2. We now know all about Firmicutes, our endospore forming phyla, and we know more about our Actinobacteria, so our Swiss cheese and antibiotic producing phyla. So we'll see you back for 10.3.